introduction. And I saw a little bit on Twitter yesterday what you did. It looks fascinating, and I wish I could be with you over there, but unfortunately I couldn't. So I'm with you in spirit, hopefully, and we'll take you a little bit into the kitchen, as we might say, going into the practicalities. So the plan for this hour would be to talk a little bit about the purpose of actually doing longitudinal work in epigenetics, because it's a lot of nuisance, it's a lot of work, it's a lot of bother. So why bother with doing a longitudinal study? The purpose should be clear. Then I will talk mostly about types of longitudinal studies and give examples first of studies without epigenetics, and then, because there are lots more of them, and then studies that included epigenetics in birth cohort studies. Then talk about strategies, some practicalities, and discuss risks and opportunities with you. Let's see how far we come. And please shout or raise your hands. I can see you in the video quite clearly if you want to ask a question, if I go too fast or too slow, or the people online use the chat function or unmute to jump in. So let's see. Why was I invited? So is there any relevant experience that that underlies my talk here this morning? Well, I'm involved together with my colleague Mirjam Oosterman in the birth cohort study Generation Square, which has been running now for a long time. It's a longitudinal birth cohort starting in the first trimester of pregnancy, and starting out with recruiting about 2,000 pregnant women, and now following these children now in their tweens. So lots of papers on that. And in the meantime, we didn't plan this at first, but yeah, if you're embarking on large longitudinal studies, you get additional ideas, additional opportunities for funding. And then later on, we also decided to collect buccal swabs in order to be able to do genome-wide analyses and epigenome-wide analyses. And these data are being analyzed now, but we don't have yet results. But this is about the actual experience that underlies some of my ideas this morning. Another part of my experience is in being a journal editor or joint or associate editor with child development, which is in our field one of the leading journals, and currently the Journal of Child Psychology and Psychiatry, which does see some epigenetic submissions, not huge amounts, but fair amounts. And my main orientation is there on mothers and fathers in the role of a parent and understanding children's well-being and development. So that's kind of a brief version of my background. Now, you know, being a citizen, you know, in this day and age requires a lot of dark humor, I guess. Being a scientist also requires a good dose of dark humor, I would say. These are just tweets of colleagues that were on display yesterday. So it's ironic or cynical almost. How can you have a career in academia, get some big existing data, never mind about the quality, just look at the variables and then think about what questions you may want to answer. analyze until you find something that you may call novel, do some sensitivity analysis just to have them, and then publish and get the PR machine rolling. And this is kind of a cynical, dark view on publication practices and research practices that people have. And of course, we are in there with good intentions, and we want to, to serve the world and society with true research. But we also are aware of all the ways in which our cognition can be biased, and that we can look at our data and see some patterns that aren't there, or think with hindsight that we might have predicted the thing that we are seeing in our data, and then kind of hypothesize after the results are known. And also another tweet from another colleague: imagine how exciting every new analysis result would be if you didn't know about p-hacking, data dredging, confounding, colliders, measurement error, and the list goes on and on and on. So kind of the fun of doing research is kind of a little bit being spoiled by all these pesky limitations that we have come to acknowledge. And so we should be aware of that, and we should try to control those cognitive biases, but also very much think about the purpose of the research. So what I will be talking about and showing you as examples of longitudinal studies, there's not one good longitudinal epigenetic study design, but there are worse ones and there are better ones. And of course, I really hope that you will be the ones also contributing to some of the better ones. So what are kind of the two broad purposes for doing longitudinal epigenetic studies? And of course, this is a gross simplification, but just for the sake of argument, we might say that one purpose of your study may be to describe or to predict. And that could be a good spending of resources to work towards precise measurement, accurate prediction, and including epigenetic data in order to improve the description of what is happening in the mother from pregnancy to birth, or in the baby in that same period. And perhaps being able to predict some later outcome. And this is highly valuable work that is sometimes being a little bit neglected or overlooked. So we want to go into the history books as the ones who have tested hypotheses and tested theories and make our fame with that. But these theories and these hypotheses, these rest on accurate and precise description. And also testing hypotheses is based on being able to accurately predict. So there's a lot of work perhaps being neglected before we go on and formulate our hypothesis and collect the data in order to test those hypotheses. And with regard to epigenetics, so there's we are learning about what you are learning about what happens in the cell, what happens to these base pairs, etc., etc. But there's a lot of things that we don't know, what goes on in the cell, in the environment of the cell, in the interaction between the environment and the processes on a molecular level. So good description of that is clearly quite relevant, and especially in the psychological and behavioral sciences, somewhat neglected. But inevitably, if you collect data, if you make descriptions, if you have hard data points, then people will want to model those data and work towards an explanation, and that is hard when you don't have all the data, which is almost never. So a brief cartoon, illustrates that. Is that of course all these data points are knowledge. You may connect them to experience. And sometimes you can project an idea on those same data points, but there is a danger of overfitting there. It's a first example of an epigenetic study that that was done for predictive purposes. Is a study done at 2 places, in Singapore and in Nijmegen. This is from the BIBO study in Nijmegen and the GUSTO study that stands for Growing Up in Singapore Towards Healthy Outcomes. And what they did is prenatally they assessed anxiety in the pregnant women, and then postnatally they collected data on accelerated aging. So they took buccal swabs and they computed from the telomere length a measure of epigenetic aging. And what you see here in the graph is the outcome of a prediction, where it's kind of complicated, but they assessed the age. And so you have the chronologic age and the cellular age, and they measured it at 2 time points, at age 8 and age 10, and then created a group high in prenatal anxiety and low in prenatal anxiety. And what you see a little bit there is a pattern in the dark colored boxes, which displays children at age 8 or at age 10 who as a fetus were exposed to a mother high in prenatal anxiety that had a little bit higher cellular aging as average for other children of that age. And so what it displays is that on a cellular level, these children looked a little bit older than what you might expect based on knowing their chronological age. And so this is an example of a study that provided a prediction based on knowledge of a psychological state of mothers during pregnancy to the extent of epigenetic aging later on in life. And the nice thing about this study is that it tested this prediction in 2 samples. So in order to be able to look at replication. And the replication was only partly successful. So it also shows us that there is high uncertainty with these predictions. So we know a little bit more, but it's only a little bit. But then a second purpose of longitudinal study, in addition to describing and predicting, could be to explain. And this is what we often are trained to see as the overall purpose of science, is to offer an explanation for the things that we see around us, and to be able to say something about cause and effect, so that we can have influence over our fate. And so we see a lot of models, and I saw them yesterday in some of the papers that you studied also, where a causal model is being fitted to a box of data. And if that amount of data conforms to a number of conditions, then we may say, well, there's something like a cause and an effect. Here's a simple model of a perinatal stressor being related to an outcome, and being related to one form of epigenetic process like methylation or telomerization. If we observe that there's an association between, for example, the stressor and the epigenesis, that we see that the stressor as an independent variable in time comes before the outcome or the epigenesis, the dependent variable, and we have controlled for potential confounders, then we may start to be a little bit more confident that perhaps the perinatal stressor may have caused individual differences in epigenetic markers, or may have caused individual differences in developmental outcomes, for example, in infants and children. But there are conditions to be met, and most studies, longitudinal studies that you will see, are designed to look at association, so expressly designed to have variation in at least the individual or the independent variables, and then you want to see also variation in the dependent variables. They will also be designed, although not always completely so, to look at temporal precedence, but the control for confounders is always a very difficult thing. And this is relevant, especially for epigenetics. I didn't look it up for telomere length, but for methylation, for example, we know that methylation at all these different sites that you can look at, on average, is partly heritable, about 20% or so. But that is an average. You don't know for the particular methylation site that you are studying, testing, or that you are that pops up from your data, whether that may also be under the influence of a heritable factor. And in the case that the heritable factor is shared between the mother and the child, then you have a potential confounder. So some of the studies that you will be seeing have tried to control for that, try to look up for the methylation sites that they are looking at. Is there evidence that these are also under genetic control? And are these genes present in the mother and in the child so that we can control for these confounders? But this is just one of the possible confounders. And there are many, many more, including the ones relating to stability in any of those measures, autoregression effects, etc., etc. So, one example of such causal inference study, not related specifically to birth trauma or perinatal mental health, but one which became quite famous, is a study that followed adults that as a child were being exposed to various gradations of wartime famine. And this was during Second World War in the Netherlands. So this is the famous famine, hunger winter study in the Netherlands, and followed them up and collected all kinds of health-related data, including also genetic data, and found a mediation effect where the exposure to wartime famine was associated with methylation on a particular site, which explained the association between being exposed to famine and later higher BMI. And so kind of paradoxical, if you are exposed as a child to the hunger winter, and then later in life you have a higher BMI, but that may be because some programming effects on the epigenome has taken place that program you during later development to to consume more calories than is good for you. So you already see some basic tools of causal modeling. So having a large collection of variables in itself doesn't say so much. So making sense of it first requires you to think about, what are in this study my independent variables, which are my dependent variables, because you want to be looking at association, and this is the first step for causal modeling. And then you may want to look at mediation. So classify your variables, not only as independent and dependent variables, but perhaps also as mediating variables. And already in the previous example, you saw that epigenetic variables are being used as mediating variables, kind of explaining the link between what happens first, for example, during pregnancy, and what happens later, for example, during childhood or adulthood. But so that answers the question how effects come about, but you may also want to answer the question, for whom is this effect occurring? So part of the causal modeling is also looking at moderators. Is this effect stronger for one group, for example, girls, or stronger for another group, for example, boys? So sex differences are often studied as moderators. We also saw that a strong tool in your toolbox should be awareness of potential confounders. Genetic ones are obvious ones, but there are many more. For example, stability in your measures is a potential confounder that should be controlled. And another tool in your toolbox and thing to think about if you are designing your longitudinal study is the possibility of bidirectional effects. Is that some of the concurrent stability in your independent and dependent variables may be explained sometimes by both of them influencing each other. But you wouldn't know if you would only have measured them each only once. So you're looking often in longitudinal design at more complicated designs in which the independent and dependent variables are both measured at multiple times, so that you can control for the confounder that independent variables kind of could have a tendency to be not only situationally dependent, but also be reflective of a trait-like characteristic. And that if you don't control for a potential cross-sectional effect, an immediate effect on the dependent variable, then you may mistakenly assume that that the independent variable precedes the outcome in the dependent variable, and that there may be a causal link there. So these kinds of models are being preferred in longitudinal designs in order to be able to control that particular confounder. And these also make sense in terms of theory. So the manifold of probabilistic epigenesis by Gottlieb almost literally displays how a longitudinal study may be designed, because that manifold is already a model that should be guiding you once you start thinking about how to include epigenetic measurements in your design. And it's kind of surprising, as we will see later on, is that there are not so many studies that have repeatedly sampled epigenetic data. But clearly, if you want to test this model of bidirectional influence, you should. To make it even more complicated is if you are interested in outcomes, in developmental outcomes in the child, going from the mother to the infant and the later child and adult, is that one thing leads to another. So we describe this as a developmental cascade that effects accumulate. This is why we are interested in these small alterations in at the DNA level, because they can accumulate into something much bigger and something much broader. So although some of the effects that you may be studying are quite small and quite weak and therefore hard to study, they may in the end be important because the effects accumulate over time, and not only within particular domains, but through interactions, through transactions, also across levels. If you're talking about BMI, you're also talking about cardiovascular health, but you're also talking about lifestyle, you're talking about movement, and etc., etc. So it's kind of snowballing effect that you are trying to measure, and that also means something for your longitudinal design, because you don't want to be too narrow in looking at different outcome measures. So what then does a basic longitudinal design look like? And yeah, so it might not be surprising. So I will show a couple of those diagrams, and hopefully for you in the back the print is not too small, but I will send the PDF of the slides, of course, to Jonathan to distribute to you later. So what you see at the top are the measurement waves, which you can count from T0 to T1 to T whatever. So the dots kind of indicate that there's a variable number of measurement waves in between. And you may, in your design, select a number of individual variables, at least one, but sometimes many. So what you see then from the top in the second row is the independent variable J, which can be a 2 or a 20. Well, how many things you can measure, but as potential predictors, but that's a possibility to do so. So, and then you have dependent variables, also indicated in a similar way. So ideally, you, in a longitudinal design, you collect independent and dependent variables at all measurement points. And then it's a truly longitudinal design. An example from our own Generation Square study we did here looked at pregnancy-specific anxiety measured in the first, second, and third trimester of pregnancy. And we also, at the same time points, we assessed state anxiety and trait anxiety. This was in a sample of about 1,000 pregnant women. And what this allowed us to do in order to test the question, how could it be that trait or state anxiety in the mother causes later pregnancy-related anxiety. You are, you are an anxious woman, and then you become pregnant, and then that anxiety feeds into anxiety about your pregnancy. Could that be the case? Or it may be the other way around, is that if your pregnancy is giving you, scary thoughts, then this may enhance your tendency to be anxious about a lot of other things as well. And what we were able to do using the longitudinal design was to test for effects in both directions. And in this case, we did find association, and we also found temporal precedence of one thing leading to another, but it was in both directions. So this is an example of a bidirectional effect, which you can only find if you do these consecutive multiple assessments. But there are also studies that look like this. You may have measured at one time point a number of independent variables, and at another time point a number of dependent variables. And these are called follow-up studies. And we will see that sometimes mistakenly people look at these designs and call them longitudinal, but they are really only follow-up. So when you factor in the birth, and this is indicated here with the orange arrow, you can have a number of variables assessed during pregnancy and variables assessed after pregnancy, of course. Ideally, everything is measured before and after birth, but this is often not possible. You cannot assess the behavior of the infant, well, only in very, very limited ways before the infant is born. And you cannot assess that there is no such thing as pregnancy-related anxiety if you're not pregnant anymore because you have given birth. So many of the common longitudinal studies are actually a mix of longitudinal elements and follow-up elements, and that is not necessarily a bad thing. An example also from the Generation Square study, where we leveraged some of the strengths of having a birth cohort in there, is that we assessed parental self-efficacy, so the trust in yourself in being able to parent well. And we assess this before birth, so asking the pregnant women, how well do you think you can take care of a crying baby? And we asked that also after birth. And then we assessed the temperament of the baby at 2 time points after birth. And of course, we couldn't do this before birth. And what this enabled us to see is that there seems to be a stronger influence of your confidence to be able to parent well on the perception of your child as being, having a difficult temperament, rather than we expected the other way around. We expected that, having a child with a difficult temperament, high negative reactivity, low soothability, would kind of eat away at your confidence in yourself as a parent, would lead to lower parental self-efficacy over time. And this is, but of course, you don't know how a perception of negative temperament comes about. And because we also had this parental self-efficacy measure before birth, so before the mother was aware of the temperament of her child, we could control for that expectations, and then saw that if you take that into account, there's really more prediction from the temperament to the parental self-efficacy than the other way around. So these birth cohort studies, they can be pretty powerful. So a pregnancy follow-up study could look a little bit like this. And an example of this with epigenetics is a quite large study from the ELSPEC cohort in the UK, which assessed prenatal depression at 2 time points during pregnancy, and then immediate at birth. Looked at methylation, and what was kind of a strength of that study. There were multiple strengths. So of course they looked at associations, which were found. There was temporal precedence, and this was also controlled because you might say, well, the methylation after birth may be confounded by continued exposure to a mother who, who takes the depression from the pregnancy into the motherhood. But in this case, yeah, the methylation in the cord blood was measured. So the exposure to a depressed mother, was almost zero. So you can really interpret this effect on methylation, and this was with epigenome-wide analysis, as an effect of the prenatal depression on these differences in methylation. But one caveat is that it was really a strength that they try to replicate their finding in an independent different study, the Generation R study in Rotterdam in the Netherlands. And then that didn't replicate. So it could have been a chance result. We really don't know. But it is a strength that they designed the study such that they could be replicating, that they could try to see whether the same finding would replicate in an independent different sample. A Norway study kind of looked similarly, and then this is one of the rare examples of studies that looked at methylation at multiple time points after birth, also looking at prenatal depression, also relating it to epigenome-wide effects in infants' saliva in this case. So here you don't have to control for the exposure to the continued maternal depression. So these effects, if there would have been effects and they didn't find any association, so it's also not confounded, you might say, but if they would have found an association between prenatal depression and methylation outcomes, it still would be difficult to interpret this as indicating a causal effect, because they couldn't control for the continued depression that mothers may take from the pregnancy into the first year of life of these infants, which may have a continuing effect on the methylation, on the epigenome in the infant. So you don't know whether any association that you might observe is already something which originated in pregnancy. Another feature of this is they called it a longitudinal epigenome-wide study, but it's really, in terms of this association, a follow-up study, because they only measured the independent variable once, and the dependent variables were not, well, they were measured 2 times, but not at the same time point as the independent variable. A study that looked at mediation from prenatal psychological state in the mother to behavioral outcomes from the US, from Conrad, looking at depression, in this case from chart review during pregnancy, and measuring methylation right at birth. So this is taking account for possible potential postnatal exposure to the mother's depression. And then looking at newborn behavior in the very first days of life. So also there you may say, well, it's plausible that these things are causally proceeding, but we are not completely sure. Of course, we would have preferred continued measures of maternal depression in order to control for those effects. And here is an example from researchers in Tilburg, in the Netherlands, looking at maternal anxiety during pregnancy, and then later on looking at methylation in 4-year-old children and behavior in the, and not newborn neurobehavior. Sorry, I didn't correctly identify this. They looked at internalizing behavior problems in these children, found some correlation, longitudinal of follow-up prediction from maternal anxiety to methylation, but not with the behavioral outcomes. So in this case, the causal model didn't quite fit. What was a strength of this study is that they included a contemporaneous measurement of maternal depression to control for the possible autocorrelation. So they did take into account that mothers may take the mental state during pregnancy into motherhood and childhood, and try to account for that effect later on. So these are just a couple of examples that I could find from longitudinal designed epigenetic studies, and mostly looking at effects from the mother on the child. And of course, you can also have many other questions that stay within the mother. But this is just reflecting my interest. And you may wonder, in thinking about your own study, so how can I collect those data? How can I conduct studies that could make a meaningful contribution to science? So I want to briefly discuss a number of possibilities to think of. Of course, if you want to look at exposure that can possibly, through the epigenome, lead to later outcome, you can use retrospective measures. The adverse childhood experiences are a famous example of that. I will return later on a little bit about the different ways into looking at this. One caveat of such retrospective designs, that you also look back in time, try to establish temporal ordering of events, is that those retrospective events may be confounded with your current state of mind, that you remember your childhood as worse than it really was, for example, or as better as it really was. And then you may get different outcomes. So usually people regard prospective longitudinal designs as better, following people over time, repeatedly surveying them or sampling them with their biological material, or doing behavioral observations using charts that are maintained for other purposes, like in the medical system, in the healthcare system, or using population-wide administrative data, all data that are being kept prospectively for the purpose of research or for other purposes, but for which you have permission to use this as a research for research. Also caveats there, of course, if you recruit people for such studies, you have to keep them with you, keep them engaged, keep them giving you their data or giving you access to their data, which can be a problem for retention and all kinds of selection biases. Still, people do this, and so cohort studies are kind of seen as kind of the standard way of doing longitudinal research. I'll give you the example of the LSPAC study, the Generation R, our own Generation Square study. There's the Gusto, Wirral, the Little in Norway, Fimbrain, and I could go on for a long time with such cohort studies. They're characterized by a fixed enrollment period. So you take a year or 2 years, and you invite as many people as possible to enroll in your study. And then within that same time space, you are administering your measurements, which takes a lot of funding, a lot of preparation time, but you obviously you can do this. Another approach, and I really want to make you aware also of this alternative approach, is to have an open cohort or a participant register. The twin registers are a very famous example of that. So people are continuously recruited as twins or as family members of twins to be part of this register, where they say, okay, I sign up as a volunteer. So if you have a questionnaire to ask me or material to collect from me, you can invite me, and then I can say yes or no. So obviously these registers have many thousands of participants. Not all of them will participate at any one time, but you are filling your database over time with a lot of variable data. So you have people come in continuously, they may also drop out continuously, but overall you are building a large data set with valuable measurements. And if you have a biobank linked to this register, then you know you're really building up something very valuable. So coming back to the issue of retrospective or prospective designs, this is also really a crucial distinction. Work of Andrea Denisi clearly shows that if you do longitudinal studies and compare what people report about their childhood looking back from adulthood, or what they report when they were children over time, then it's clearly not the same. So remembering that your parents have separated, that is kind of a large overlap from between the retrospective and the prospective measures that are included in longitudinal studies. But saying, you know, I have been maltreated as a child is very different from prospective recording of maltreatment followed over time. These are different people who are observed to be maltreated, or who say as an adult, I have been maltreated as a child. And the same goes for sexual abuse, physical abuse, emotional abuse in particular is something which is very, very difficult to find alignment with. These are really, really different groups who report emotional abuse. And this is something also to take into account if you're thinking about birth trauma. Are you looking at this as an objective event and following this prospectively? Or is it something that you assess retrospectively? And these can be very different people with very different outcomes. So also, so for the field of child maltreatment and adverse experiences, Denise says, well, we should be looking at and asking our questions in terms of you know, who is traumatized, and the answer on that question depends on the method. So, you know, trauma by giving birth, who experiences birth trauma, you get different answers with prospective or retrospective measures. How has it impact? You know, so one of the things to look at is there also pre-existing liability? Are some people more prone to experience birth trauma or not? And can some of these liabilities perhaps explain some of these later effects? And then which mothers and children? Because most children and also adults develop psychopathology, you know, without being exposed to maltreatment or abuse or birth trauma. So, you know, so asking yourself, you know, which people are seeing the effects of those events may be one of the important scientific challenges here. Just some notes to almost finish off by saying, you know, in terms of your strategy of going about conducting longitudinal science studies, we have learned that it's very important to include stakeholders. It can also give you a stronger license in order to, you know, be asking for the time and effort of people to contribute to your research, but also a lot of wisdom and insight and creativity they can add to the process. Another important advice, I think, is to align your design and also your measurements with other designs and measurements that are used by other studies in the field, so that you can also combine and replicate. So join consortia or look at independent replication across studies, which is more informative if your measures and designs are more similar. Building biobanks, so collecting perhaps a little bit more biological material than you perhaps need at first, because some of the methods may change. So people who looked at candidate genes in the past, they may be very happy if they have kept some of the genetic materials when they found out that candidate gene approach is really not viable anymore, and perhaps genome-wide analysis or epigenome-wide analysis is the way to go. But you cannot go back if you don't have the material anymore. Joining consortia is, I think, really a very important issue to consider. This is an example of a consortia that pulls data from now about 39 different longitudinal cohort studies that all use the Illumina 450K chip for looking at the epigenome. So if you have included and if you have analyzed your material with that kit, you can become part of that consortium and publish as part of a much larger group, increasing your precision and increasing your replicability. Which makes it also all the more important to get, you know, the different researcher degrees of freedom under control. So many of these consortia and also many of these cohort studies, if you want to use the data, also require already that you pre-register your hypothesis and your analysis plan. There are multiple ways of doing this. And also to take into account the statistical power. You know, if you are still planning to see how many people should I recruit, then of course that is important. But also if you have already recruited a particular group, then you may ask yourself, you know, how strong does the effect need to be in order to have sufficient statistical power with the sample size that I achieved. So you're doing sensitivity analysis, which can help you to decide, whether this effect size crosses certain sensible thresholds that would make the study even worthwhile to do as a standalone study. But obviously, if you are planning to be part of a consortium, then even small samples can be useful and helpful because they contribute to this greater whole. And therefore, I cannot recommend enough the recommendation at the very right on the slide, to include opt-in consents for data sharing and data linkage with your participants. And this is not trivial, but your life will be undoubtedly be much more happier if in your informed consent you have included these clauses and enabling you access to these larger data pools. So yeah, what are good ideas to finish off and bad ideas? I think going alone in your studies is not the best of ideas. Going together provides enormous opportunities, which of course can only happen if you orient yourself well in the field and see what other people are doing. Putting all your eggs in one basket could be risky, which could mean that you hold out some of your data or hold out some of your biological material and include multiple operationalization, but also include multiple theories, so that you don't depend on one particular hypothesis in order to make your study as a success. And design your study to be also informative if the effects are null. And once we start implementing stronger controls for confounding, we will be expecting many of those epigenetic effects or mediation effects to be reduced to null, to zero, which is not informative if your study is very low powered. So if you design your study so that you can do equivalence testing or other tests that see, is the effect lower than negligible, then a study with no effects can still be very informative and very helpful because it helps us to start looking elsewhere in order to do more informative research. But that also will require larger samples and thus collaboration. So I'm aware that I've been talking solo, so going against my own advice to be kind of solistic. But Jonathan, is there time?
We'll always make time for questions, Carlo. Always make time for questions, because that was, I think everybody would agree, really cool as a lecture. So thank you very much. So I've got the microphone.
I see hands clapping. I don't have any sound, but
you didn't have the sound? Well, sorry, but you did get a very good round of applause. So anyway, I have the microphone, and I'm now just going to hand it to whoever sticks their hand up for a question.
It seems I lost the sound.
The microphone is on. On channel 2, it seems to be working. Yes, it is. I can see the green moving on the... Well, we have a question coming. Maybe, can you type the question into the chat for Carlo? Yes?
Yeah, my sound is now back. Sorry, I lost...
My question is, maybe it's a little bit out of the park, but I'm listening and looking at all these different studies that were done, and they all concern the mother and the way that the pregnancy affects the baby, and that's clear. I can understand that. But have there been any studies done on the state of the father at the time of conception or preconception, and then seen what the difference is with the child are there. Kind of seems like we're putting all the onus on the mother, and we're not even considering the fact that maybe the father has something to do with the epigenetic changes.
Yes, that's right. Well, my colleague, Marjan Bakemans Kranenburg, with her PhD candidates has been conducted many studies on fathers also during the transition from to parenthood. And she has been looking to also effects of some interventions you can do prenatally in order to influence outcomes in parenthood and then outcomes in children. And I didn't talk about those studies because they are intervention studies, and these are of course great ways in order to arrive at causal inference. But one of the things that she has been studying is to do prenatal video-based feedback. So using the sonograph in order to stimulate interaction between the expecting father and the fetus, because fetus responds to mild touches and manipulations of the belly, and also sounds. So she had the expecting father sing to the fetus, and provided feedback through video on that to see to what extent this may be related to later outcome, including oxytocin responses in the fathers and different neurobiological outcomes in the child. So obviously that work is trying to understand also what also happens with the father, psychologically, but also neurobiologically. And some white matter scans in the brain have also been conducted in that particular study to see to what extent, for example, recollection of abuse may be related to differential responses that expecting fathers are giving to infant-related cues. So we clearly are also looking at the side of the father, and we also know from attachment research, a recent study that we did with Or Dagan, collecting whole family attachment data with mothers and fathers from around the world in an individual participant data meta-analysis, is that there's no hierarchy there. So quality of attachment with the mother and with the father has similar predictive power for social emotional outcomes in the child. It's not that security in the relationship with the mother kind of trumps insecurity in the relationship with the father, or vice versa. So clearly, in terms of biology and neurobiology, there are some very particular things that are happening with pregnant women and delivering a child, and pathways of influence that just are not present for fathers. But fathers go through socio-emotional and psychophysiological changes as well, and we are beginning to understand how that may also be channeled into interventions that could help the family as a whole.
Okay, are there any more questions? Do you have a? Oh, there was a question in the chat. It was just what do you mean by state and trait anxiety?
Yeah, so I'm just typing the name of the colleague who has done the father study. So trait anxiety is the description that you give of yourself as being generally prone to anxiety, whereas state anxiety refers to the extent of anxiety that you have reported over the past week or so. And so state anxiety may be much more related to your situation. For example, if you're in a bad situation or under threat, whereas trained anxiety describes something that, characterizes you from birth, like a personality or temperament characteristic.
No, I'm just going to come in at that point because I had one of the psychologists I work with, Klaus Vogler, just say to me, because he explained this to me once, in fact, your state anxiety is now. How are you now? And your trait anxiety is globally. What's your personality? Are you somebody who's generally laid back? That's how he explained it to me. And I think that's a very good simplification of it, in less technical words than what Carlo said.
Yes. And, in the audience is Annalena Holopainen, who actually also looks at parenting stress as something which may have a trait component, which may be the largest part, and a state component, which may be more dependent on the situation. So it's very interesting to see in her study how that plays out in terms of prediction for other outcomes.
Okay, so that brings to a close, I think, the lecture from Carlo, which is now absolutely the perfect introduction to the next session.